Okay, next we're going to be taking a further look at the microscopy of skeletal muscle cells. What you see here in this image is the sarcolemma. That is the clear membrane that surrounds the structures that are on the screen. The sarcolemma, again, is the plasma membrane for a skeletal muscle cell, or really any muscle cell. Inside of this skeletal muscle cell, you're seeing multiple rod-shaped structures. And one of those rod-shaped structures is pulled out so you can get a better view at it. Each of those rod-shaped structures is called a myofibril, and myofibrils are filled up to the brim with myofilaments. There are two myofilaments, actin myofilaments and myosin myofilaments. And these are going to be really important for skeletal muscle contraction. The myofilaments in myofibrils are regularly arranged to create light and dark bands that give skeletal muscle and cardiac muscle their striations. You can also see a nucleus in this image. Skeletal muscle is nucleated over and over, so it's multinucleated. You can also see green T-tubules and blue sarcoplasmic reticulum. Now the blue sarcoplasmic reticulum is only shown a little bit, but it actually is located around each myofibril, just like the T-tubules, also known as transverse tubules. The ends of the sarcoplasmic reticulum in blue that are adjacent to or abut the green T-tubules, those are called terminal cisterns. And two terminal cisterns plus one T-tubule is known as a triad because that's three structures together that meet at a location around the myofibril. So that's basic microanatomy for a skeletal muscle cell. Let's look closer now though at the striations. When it comes to the striations, we have a dark A band and a light I band. The A band is dark because of increased overlap between the actin and myosin myofilaments within the myofibril. The I band is light or appears lighter in the sarcomere or in skeletal muscle fiber because there is decreased overlap of actin and myosin myofilaments within the myofibril. When we're talking about myofibrils, again, we're talking about those densely packed rod-like contractile proteins that make up about 80% of the muscle volume for a skeletal muscle cell. And they're arranged in that regular pattern, meaning that the overlap or organization of actin and myosin myofilaments creates these striations. Here we can see one myofibril. Inside of it is a dark red line, actually multiple, and multiple dark blue lines. Areas where you see the red and the blue overlap are going to be the A bands or the dark bands. Areas where you see just blue fibers, those are going to be the I bands or the light bands. The blue fibers are actin and the dark red fibers are myosin. Another name for the actin myofilament is the thin filament, and another name for the myosin myofilament is the thick filament. And we're finding them within this myofibril. They are themselves the myofilaments, but they're arranged in a way where they're segmented throughout the myofibril. And each segment from Z disc to Z disc is considered a sarcomere. And remember, a sarcomere is the smallest contractile unit of a muscle cell. Here we've pulled out just the myofilaments. So now you have a better view of the thick filament or the myosin myofilament and the thin filaments or the actin myofilament. We've pulled them out just from Z disc to Z disc, which is the outline of a sarcomere, the smallest unit of contraction. And you can see there's areas where the two myofilaments, actin and myosin, overlap. That's the dark A band. And areas where they don't overlap are the light I bands. Areas where the thick filaments meet in the middle, that is going to be the M line, and it's found in the H zone. And then we're also going to have other structures that we can pinpoint, and a way to kind of remember them is to come to this slide, where we really go over sarcomere structure. First, the A band is the dark band that contains thick filament with an overlap of thin filament. The I band is the light band that contains only thin filaments. The Z line, or Z disc, marks the boundary between adjacent sarcomeres, and the M line is the connection in the middle of the H zone where we see thick filaments connected to each other. And the H zone contains only thick filaments, and it's in the middle of a sarcomere. And a zone of overlap is where thick and thin filaments overlap, and generally is also described as the A band or the dark band. So now that we've looked at this anatomy for a sarcomere, let's also look at the structure of each myofilament. We'll start with the thick filament, or the myosin myofilament. They're made up of the protein myosin, and the myosin molecule actually is going to have two globular heads and then two rod-like tails that wrap around each other. Each globular myosin head has a binding site for ATP. And in order to create the myosin myofilament or thick filament, you have multiple subunits of myosin bound together. So you have this really thick rod that has multiple myosin heads laterally positioned around it. Then we have the thin filament, or the actin myofilament, which is composed chiefly of the protein actin. Actin subunits are globular. They contain an active site that can bind to myosin heads, and that actually would form a cross bridge during contraction. But there's not only actin in the thin filament. There's also tropomyosin and troponin. Both of these are regulatory subunits, so regulatory proteins, and they're actually bound to actin. And when they don't bind calcium, they block the myosin head on the myosin thick filament from binding to the actin active site. So as long as troponin and tropomyosin are arranged around the actin active site, you can't form a myosin-actin cross bridge. So it doesn't matter if contraction could happen, it can't with troponin and tropomyosin in the way. You can see in the image that the troponin complex is more of a globular complex, and the tropomyosin complex is more of a rope-like complex. Together, they block the actin active site from binding to myosin. But if we could get calcium, calcium would bind to troponin, which is connected to tropomyosin, and would rotate out of the way, and myosin could bind to actin and initiate contraction. Let's move on and talk more about the T-tubules and the sarcoplasmic reticulum, because these are two sets of intracellular tubules. They participate in regulation of muscle contractions. T-tubules are extensions from the sarcolemma, so they're not only going to be connected to the sarcolemma, they're going to increase the muscle fiber surface area. So they're also continuous with that extracellular space. They're going to be very beneficial for getting an action potential that arrives at the sarcolemma and getting it to travel inside of the cell, into the T-tubules, because they're connected. Then you have the modified endoplasmic reticulum, known as the sarcoplasmic reticulum, and it's going to be found around every myofibril. And its role is to regulate the intracellular levels of ionic calcium. It's going to regulate how much calcium is in the sarcoplasm. And that's actually where we store calcium. In skeletal muscle cells, calcium is stored in the sarcoplasmic reticulum. So it can store and release it on demand when the muscle fiber is stimulated to contract. That makes calcium the final go signal for contraction. Here we can see the triad relationship that T-tubules and the sarcoplasmic reticulum share. Now on each side of a T-tubule, you have the ends of a sarcoplasmic reticulum against it or abutting it. The ends of a sarcoplasmic reticulum is called terminal cisternae. So two terminal cisternae next to a central T-tubule make up three structures known as a triad. They are responsible for a step in skeletal muscle contraction called excitation-contraction coupling. So together, these structures are going to be able to regulate muscle contraction. So how do the T-tubules regulate contraction? Well, ultimately, muscle contraction is controlled by a nerve-initiated electrical impulse. So we need a neuron to communicate through a series of steps, an electrical impulse that arrives at the sarcolemma. Once it arrives at the sarcolemma, it can travel from that plasma membrane to the T-tubules, because the plasma membrane and T-tubules of a skeletal muscle cell are connected, and into the deepest parts of a muscle cell. And that is going to get to every single sarcomere of a myofibril because the T-tubules are wrapped around the myofibrils. And they're also going to be connected to the sarcoplasmic reticulum that contains calcium. So this electrical impulse that goes all throughout the cell can also cause the release of calcium, meaning the T-tubules are really acting like a rapid telegraph system to get us the final signal for contraction, calcium, and that would ensure that every myofibril in the muscle fiber contracts at the same time. So before we move on to the next video, let's review the functional organization in skeletal muscle, and we've touched a little bit on some of the physiology, so I'll try to mix that in. So first, when we're looking at skeletal muscle as an organ, there are 3 layers of connective tissues. From the outside, epimysium, middle, perimysium, and inner endomysium that help us organize skeletal muscle. Perimysium is going to be that middle covering that surrounds bundles of cells called fascicles. Those bundles of cells covered by endomysium contain within them many different myofibrils. And myofibrils are rod-shaped proteins that are covered by T-tubules and sarcoplasmic reticulum. Now the sarcoplasmic reticulum holds calcium, the T-tubules are connected to the plasma membrane, and together they help regulate muscle contraction. They can do that because they're wrapped around these myofibrils, which are made up of actin and myosin and myofilaments, in a regular order, creating structures called the sarcomere. The sarcomere is the smallest unit of skeletal muscle contraction, and the myofilaments are arranged in overlapped areas and non-overlapped areas, with the overlapped areas producing a darker shadow and creating the A band or dark band, and the non-overlapped areas producing less of a shadow and lighter bands called the I band. So that's our basic anatomy with a little bit of physiology thrown in for skeletal muscle, and we'll go further with the sliding filament model of contraction in the next video.